I know though.
Okay, good morning. Let's have a class. And we will talk lots of the chemistry today and trying to finish this section as much as we can. Okay, some of the announcements under the plan. We will have this week, today we talk about biochemistry. Thursday we're going to try to go to the DNA replication. And the 4th, November 4th, we want to talk about transcription translation. November 6th will be a guest lecture for Dr. Scott Baldridge. Because I'm going to University of Connecticut to join their PhD defense, and I have been invited to talk there. November 6th will be Dr. Scott Baldridge's special talk about parasite. I'm offering 5 points, extra points for attending this guest lecture. So please come in. Okay, especially you didn't do very well in the exam 1 and the exam 2. So 5 points for the attendance. When we came back, hopefully this will be lecture operant, then the 13th will be the review, and the 18th, November 18th will be the exam 3. Okay, so that's basically is a procedure, is a schedule, how we're going to talk about. Okay. I'll skip these, I'll come back to later on. So we're going to talk lots of the chemistry today, lots of the chemical reaction. Basically go over glycolysis, citric acid cycle, and the fermentation. So you have these slides. We talk about all the bacteria. All the bacteria growing in the lab is chemoheterotrophs. That is their very general name. And you have like a chemoautotrophs, like a different type of the name, which means a different combination of the energy. We couldn't skip that. Okay, this is what we talk about the first slides and second slides. Talk about this combination with carbon resource, energy resource, electron resource. We just skip that. It's a little complicated to talk right now. We'll go back later on. So what we focus today is chemoheterotrophs, which means general bacteria growing in the lab. We're going to talk about aerobic respiration and the fermentation. Anaerobic respiration will be very briefly. So we have these slides here, and also we have these slides. What this means, this is very important, what we talked already, and do some of the conclusion. And I'm not going to go into do the review of the first lecture, but we will review when we talk about today, when we go those all details chemicals. Okay, so first of all, there is a conclusion, which is a key concept to talk about energy transfer, which means if electron donor is organic chemical, and electron acceptor also organic chemical. This is called fermentation. Using electron to represent the energy transfer. So if electron donor is organic chemical, and electron acceptor also an organic chemical, this is called fermentation. If electron donor is organic chemical and electron acceptor is inorganic chemical, and this is called respiration. So that is the first difference between that. What is called a fermentation, what is called a respiration. Now, if the electron donor is organic chemical, electron acceptor is inorganic chemical, it is oxygen, which means the final electron acceptor is oxygen. We call aerobic respiration. If the final electron acceptor is inorganic chemical other than oxygen, for example, sulfide, this is called anaerobic respiration. And you will find during the exams there are some questions asked you about that. And this is the best definition for fermentation and respiration using electron transfer, because coupled with energy transfer. Here I have another thing I want to talk about is during the electron transfer we need an electron carrier. For example, NAD plus nicotinic adenine dinucleotides. These type of electron carrier, they must be recycled, which means you see that this guy must be free available in the cell membrane transferring system. If we don't have this one available, bacteria cannot survive. And you can understand from the chemical structure is NAD have two wells will be accepting the proton. And this is an empty vessel. It's an electron carrier, which means something is ready to go. If we do not have NAD plus as an electron carrier recycled in the system, the bacteria will be dying. So this is from the beginning what we want to talk. It's a key concept here. And the slides here and this slides is the same thing. We do the conclusion. It goes from here. So what is talk about fermentation? What is respiration? What is aerobic respiration? What is anaerobic respiration? Is right here. Okay? Plus NAD plus has to be recycled. So that's what the slides here and talk about. So what is the outline of the class? The outline of the class is coming from glucose going through a cycle called glycolysis. And then we'll generate a product called pyruvate. And pyruvate, we're going through citric acid cycle. This will be maximize their energy production, which is ATP. And this is also could go sideways, which is going through fermentation. So today, we want to folks talk about every detail of the glycolysis, citric acid, and the fermentation. Now, go here, there is a side chain here, which is called pentose phosphate. We will very briefly mention at the end. We want to go detail about that. Okay, so this is pretty much the outline. So we want to talk about everything very detailed. What are they? A little bit concise in biochemistry class, but more is focused on the energy production, how they generate. Let's talk about the fermentation first. And we already said, what is fermentation? Is electron donor is organic chemical, and electron donor is organic chemical, and electron acceptor is also an organic chemical. So, what is a good example? A good example is pyruvate reduced to lactic acid. At the same time, do not forget something happens, is NADH2 oxidized become NAD plus. So what are they? That's carboxy acid, carbonyl group, and CH3. Then go into lactic acid. That is lactic acid. And coupled with NADH reduced to NAD, oxidized become NAD plus. Okay, now, the key thing is here. Where does oxygen go? Oh, sorry, where does H2 go? Of course, it is added here. It is added here. That's why it showed up. So you understand, it's very simple. It's a pyruvate or pyruvic acids reduced become lactic acid. At the same time, NADH2 oxidized become NAD plus. So therefore, there is some question will be generated. What is NAD plus here? This is a oxidized product. And this is reducing product. We also call, if pyruvate become lactic acid, this is called homo fermentation. And this is also fits the definition organic chemical and organic chemical. Now, we had a homo fermentation. In other words, we will have hetero fermentation. What is that? That is pyruvate reduced to acetaldehyde and further reduced to ethanol or alcohol. So what is that? What's the structure for this guy? This is pyruvate. Here, lose carbon dioxide. Once you lose the carbon dioxide, what this means, this is gone. So, this becomes, this is aldehyde. Okay, now, goes here, what's going to happen? Again, NADH2 oxidized become NAD plus. Okay, what are these goes in? What are these H2 goes in? Of course you're going to be added. Add here, so become what? That is ethanol. During the fermentation, if you created some organic chemical other than lactic acid, we call hetero fermentation. Okay, some common knowledge. If this goes further, what will become? If it keep going, what going to be? What is this? Acetic acid. What is that? Vinegar. So, the easiest way to making a vinegar is you have a wine bottle and you open it or you loosen the cap, put on your dome for 3 months. Not even 3 months, about 1 month. Then you taste it, it's acid. It's vinegar. Obviously, it's not edible. Okay, just a chemical reaction. That's acid. and acetic acids. This is called secondary fermentation. We don't want to talk about here. This is for chemistry we're going to talk about. Here, we stop here, is ethanol will generate. Okay. So these are the two examples, which is explain homo fermentation and hetero fermentation. Now regarding the fermentation, there is some of the conclusion. Number one, how many ATP generate? No matter how complicated it is, only 2 ATPs generate. Now, people will say, you only have 2 ATPs generate. How bacteria can survive? Can still survive? The reason is I just said. You have this guy keep showing up. Because you have free available NAD+. That's why fermentation can keep doing, although it's only 2 ATPs. And these 2 ATPs generate, it is coupled with glycolysis. And where it is, right there. Can you see on the top? Glyceraldehyde 3-phosphates oxidized become 1,3-biphosphogycerate. During that, you have the connection, which is NAD become NADH2 and do the cycle. And the pyruvate become lactate. They are coupled with glycolysis. And that's what the NAD is recycled from. And why the bacteria can survive. That's the reason. Okay? And where are those 2 ATPs created? Through substrate level phosphorylation. Okay, I use P circle represents phosphorylation. So this is only through substrate level phosphorylation. Okay, so very simple. This is for fermentation. Of course, fermentation is anaerobic, which is without oxygen. Okay, otherwise it will cause aerobic respiration or anaerobic respiration. It's without oxygen. This is the one pretty applicable, which is from the application standpoint, talk about what is fermentation. What's the applied science? So glucose generates pyruvic acids. Then pyruvic acids with different bacteria, in the absence of oxygen, they could generate these products. So for example, if the oxygen, if the microorganism is probionic bacteria, they will generate probionic acids and carbon dioxide. And this is making Swiss cheese. And you see those holes there. That is carbon dioxide. That's why you see those so many holes there. And you feeling the Swiss cheese taste acid because probionic acids. Okay? This is something else. That's probionic acids. Folic acids, acetic acids, and probionic acids. Acetic acids is actually a very short chain fatty acids. Very short chain fatty acids also could be antimicrobials. That's why acetic acids is antimicrobials. Okay, so something else. If the organism is aspergillus, which is a fungi, or lactobacillus or streptococcus, and this streptococcus here is not a pathogen, it's streptococcus thermophilus. The species is thermophile, it's a beneficial bacteria. It will generate lactic acid. And this is for cheddar cheese, soy sauce, and yogurt. If the Saccharomyces, and this is the first slides, very first slides you did in our lab section, the number 1 lab. will generate carbon dioxide and ethanol. This is hetero fermentation. We'll have wine and the beer. Of course, beer, you have a lager and you have an ale, which is talk about top and the bottom fermentation. And wine, you have a red wine, white wine, you use a grape and you're going to be crush it. These are the other classes we will talk. Okay, so we just be very briefly, we have a carbon dioxide and ethanol going to generate. Okay, you can buy a bottle of a nail polish remover. The components is acetone and isopropyl from Ultra or Sephora in the store. It's quite expensive. Sometimes $10, $20 for one bottle. Where they come from? They come from Clostridium. And remember, Clostridium is a strict anaerobic bacteria. So without oxygen, they of course can do fermentation. They will generate acetone. They could be using for a nail polish remover. And the last one, this is what I talk about, is E. coli. This is the E. coli 9037. It's a beneficial bacteria, an acidobacter. They will create acidic acids. And that is with secondary fermentation. Acidic acids, if the components is about 5% and the concentration is about 5%, it is vinegar. Okay, so this is a very good slide when we talk about how the application of the fermentation during the manufacturing of different food products. So that's what we talk about for the fermentation. Okay, so fermentation, quite straightforward, but they still have a lot of chemistry. So we remove these parts. We're going to move to the next one is a big chunk. We're going to talk about glycolysis and the citric acid cycle. And this is called aerobic respiration. We will talk about every detailed chemical structures and also how we calculate the ATP. And that will be the key. So we want to spend in time talk about that. Pretty much it's go over your biochemistry class, but we have a little bit of difference. We want to talk and we also want to emphasize. So we will remove the whole blackboard, have a larger space, we can talk about that. So, the outline here, at the very beginning, we have a glucose. And glucose will go here, talk about generate pyruvate. And this whole thing we call glycolysis. So first of all, the glycolysis what we talk today is what you learned in the other classes. It's a typical cycle pathway, branch pathway, called Ebden-Meyerhof pathway. This means the generally bacteria, vegetative bacteria. Soil bacteria is different. So we have some students from plant science, soil microbiology major, although it's not many. Soil microbiology, glycolysis is different. They are going through Ebden-Dorohof pathway. It's a completely different pathway. So we're not going to talk about here. What we here talk about is still a very typical Ebden-Meyerhof pathway because it's generally for E. coli and all those vegetative cells except the soil bacteria. Okay, what is that? Let's talk about it. The first area is from glucose to the intersection will generate fructose 1,6-biphosphate. This area, we are using ATP, so we call it an investment phase. This area totally is used to ATPs. Right very from the beginning to the intersection of the branch. So let's talk about that, all these chemicals, what do they look like? We've got to draw here, okay? This is a glucose. So you should know how to draw glucose. Very familiar. And I hope in your chemistry class you already learned about it. Because this one is not difficult. So 6-carbon sugar, glucose. This is what we call the Haworth configuration. You have open rings and the chair configuration, Fisher projection, and the chair configuration. That's the other things. This is a Haworth configuration. And that is glucose. Our most common 6 carbohydrates we're using available in the world. The first step, we're going to add a phosphate. So become, this is the same. So you can just do the same thing. What we call this? Glucose 6-phosphate. What means 6-phosphate? Which means a phosphate add onto the carbon number 6, add right there. So becomes glucose 6-phosphate. I use P, circle represents phosphate. Okay, what is the enzyme for that one? And you learned in your biochemistry class, this is called hexokinase. Is that correct? Yes. But in my class, this is for fungi. And we are talking about is animals, human beings in your biochemistry class, and for the fungi is hexokinase. If for bacteria is not, they are through group translocation. If you still remember. And we said that it needs to have phosphoenolpyruvate at the end to help. So for bacteria, they are using group translocation. And for fungi, very similar to your class, called hexokinase. So that's, we can say it's for fungi only. Yeast and molds. Okay, is that a hydrolyzed ATP? Of course, you add a phosphate there. Where that phosphate comes from? That is ATP become ADP. That's why that phosphate is released. So this is an area we have to circle. And that is 1 ATP used. Okay, glucose 6-phosphate, the next one. It will become, we call fructose 6-phosphates. So what do we call this? Glucose 6-phosphates become fructose 6-phosphates. When we draw the sugar, it's a little difference. What are the difference? Look at that. And compared to this. So what's the difference? CH2OH, this is OH, so you go here, H, OH, OH, H. And remember, this is H. This is not OH. Okay, CH2O. What's the difference? Chemical numbers are the same. The structure is changed. What is the terminology for this one? We call chemicals, numbers, components are the same, but the structure changed. What's the terminology for this in the chemistry? Called isomerization. So the enzyme is called isomerase. But of course, you want to say the detail, you can say phosphor, glucose, isomerase, blah, blah, whatever, but it's isomerase. Okay, this is nothing happened, just change the chemical. Now, why they do this? you always remember, in the chemistry area, they need the morphology is symmetric. Then we can keep the balance, like the human body, looks like symmetric. So when you have here, of course people are going to add something there. That's why they can survive and keep the chemistry during their evolution. So the next one, it is important, but it's easy, you add another stuff there. Okay, CH2O phosphate. Then you go here, OH, H, OH, OH, H, H, CH2, O. What is called? This is carbon number 1, this is carbon number 6. So we have fructose 1,6-biphosphate. What is this called? What's the enzyme? Well, it's just kinase, like phosphofructokinase. That's just some common name. Does the ATP used? Of course it's used ATP, otherwise where this comes in? So this is another place which is ATP hydrolyzed become ADP, and therefore it's used, not created, 1 ATPs. So how many ATP used here? -2. Yeah. They used. And the product's called fructose 1,6-biphosphate. Right here. Okay. Okay. Once this chemical comes out, what did you find? That's a very balanced structure. Once it's balanced, what people want to do? Cut it in half. That's what it is. Life cycle is very magic. They keep order, disorder, order, disorder. When you see like this, they want to make it symmetric. Once the symmetric comes, they want to cut. It's always like that. You don't have money, you want to get the money. When you get the money, you want to use it. It's the same thing. You want to cut it. So when you cut it, it becomes what? Glyceraldehyde 3-phosphate. So let's go here. It's going to become branch, okay? Because when you cut it, you have 2. And I'm only going to talk about 1. The other one, I'll write chemicals there. So this will become glyceraldehyde 3-phosphates. CHO. That is easy. Aldehyde. CHOH. CH. That is called glyceraldehyde 3-phosphate. Okay, this is something we want to say. You should learn your chemistry. This is the simplest sugar, called aldose, which means 3-carbon sugar. So, once you break down, goes here. What's the enzyme called? Aldolase. Six-carbon sugar becomes 3-carbon sugar, but you have no phosphates. So I call it glyceraldehyde 3-phosphate. Now, glyceraldehyde 3-phosphates, they also have a transition step. This is something additional. It's called CH2OH, then CH2O phosphate. That's called dihydroacetone phosphate. That's dihydroacetone phosphate. This is nothing really special. It's just to keep the balance in their life cycle. It's a side chain reaction. Okay, nothing really happened. It's just chemical changes. So glyceraldehyde 3-phosphates. By the way, just give it to you. Okay, now what happened? Glyceraldehyde 3-phosphates, when something like this, if you know the chemistry, this energy for those OH, which is the hydroxy bonds in a C end, it's very weak. It's going to be modified by something. Okay, that's called the principles of biochemical reaction. We don't want to go detail about that. So what we do? We have COO phosphate comes, and this will be using our phosphates. Okay? Middle, we don't change it. HOH, CH2O phosphate. What is called? 1,3-biphosphoglycerate. Okay, something very important here. 1,3-biphosphoglycerate. Where this guy goes? Some residual phosphates in the cell membrane transfer systems is provided. At the same time, NAD reduced to NADH2. This will happen. So give you 1,3-biphosphoglycerate. Now, did you notice that? This is gone. That proton is gone. So what's the enzyme we needed? Dehydrogenase. So the enzyme called dehydrogenase. Okay, is there ATP created? Yes, we'll talk about later. Okay, we just skip that. Dehydrogenase. 1,3-biphosphoglycerate. Well, you have 2 phosphates there, they're ready to go. We don't need that much. Like I said, no balance, you balance, you want to use it. So then you use it. So next one. Goes this. What is this called? This is called 1,3-biphosphoglycerate. So that is called 3-phosphoglycerate. Okay, then what does that one go? Well, we have to use it. So, ADP with these phosphates generated ATP. What is the terminology for this one? What's the terminology for the ADP created ATP here? We learned called substrate level phosphorylation. Why we can do that? And we can review what we learned in our lecture 1, because the phosphor energy transfer power of 1,3-biphosphoglycerate is 61.3. How much is here? 30.5. And the energy transfer power is much larger than ADP becomes ATP, or the opposite way. Therefore, this will be an exogonic reaction. So that's why substrate level phosphorylation will happen. So becomes 3-phosphoglycerate. Okay, what's the enzyme for that? It's called kinase, is that right? Yes, very common name, called kinase. Okay, once you have a 3-phosphoglycerate, what will be? Go down. 3-phosphoglycerate, this move to the middle. There's not much to talk. You just move to the middle. COH. Phosphate. What this called? 2-phosphoglycerate. What's the enzyme for that? This enzyme varies, a function is similar to isomerization, but we don't say it. We call mutase. So the enzyme, we call it a mutase. Then become 2-phosphoglycerate. Okay, 2-phosphoglycerate. We're going to go a little bit on the here. 2-phosphoglycerate comes. What are we going to do? What are we going to do? 2-phosphoglycerate comes. We have something going to lose water. We call it enorization. So, this enzyme called enolase. And you should know the terminology happens in biochemistry a lot called enolization. This is equals lose water. This is gone, this is gone. So what it looks like. Then what's that? We talked already. Phosphoenolpyruvate. This is very important for the bacteria because this guy gonna go back here to do group translocation for bacteria to transfer phosphate. Okay, to let glucose become glucose 6-phosphate. So phosphoenolpyruvate. Is there anything happen for the ATP? Pretty much not. Is that right? We don't see anything. It's enorization. It's basically is you are lose water. Now what are we going to do? That's pretty straightforward. Become a pyruvate. Now where is this guy go? Obviously goes to ADP. So ADP with a phosphates becomes ATP. What's the enzyme? Kinase. So that's a pyruvate generator. So how many ATP created? It's a very tricky question. 2 ATPs multiplied by 2 equals 4 ATPs because it's 2 lines. And you use 2 ATPs here. So glycolysis, how many ATP? Minus 2 ATPs, add 4 ATPs, equals 2 ATPs. But I want to tell you something little different, little tricky maybe. Glycolysis generated 2 ATPs in the biochemistry class, which means it is a closed cycle, which means it's individual. It's not created to part, it's not a part of the aerobic respiration. If it's part of the aerobic respiration, this guy can be calculated. So you understand, when we talk about glycolysis individually, like what we talk about here, we created 2 ATPs. We are talking about is glycolysis is an individual closed cycle. Or you can understand that we are talking about the glycolysis is coupled with fermentation. This is key. That's why 2 ATPs. And the biochemistry book think you're smart, so they skip it. But if glycolysis is part of the aerobic respiration, ATPs it is. NADH reduced to NADH2. When I was student, it is 3 ATPs. And you learned 2.5. Let's just do 3, okay? It's easy. Here are 3 ATPs. So how many? That will be minus 2, add 4, add 3 multiplied by 2. So how many? It's 8 ATPs. Okay, clear? We have to clean some of the concept. If it's part of the aerobic respiration, this, you have to mark it. So it becomes 8 ATPs. Now, what's the terminology for here to create ATP? That's called oxidative phosphorylation. We have a theory called chemosis by Peter Mitchell, a great British scientist in 1932, to talk about the mechanism behind it. We already talked about it in the lecture on Tuesday, or last week, okay? So that's part of the end. So we finished about here. Now we're going to talk about the cycle. Okay, so let's do the cycle. Pyruvate. We have a big chunk of the blackboard here, so that's good. We can use it to talk about the citric acid cycle. So we talk very top. This is pyruvate. First of all, pyruvate going through a transition step, then become COOH, C2, SCOA. This is called acetyl-CoA. I want to say this, acetyl-CoA in the biochemistry book, it is a very acronym. This is not right like that. This is much more long chain. They just be called acetyl-CoA to let you understanding, okay? And here what happened is NAD reduced to NADH2. Okay, now the acetyl-CoA gonna go through the cycle. This cycle called the citric acid cycle, so let's do it. If we want to know the chemical structure of citric acid cycle, you first of all, obviously, you need to know what the citric acid looks like. So, let's first of all, you have to know what is citric acid. Citric acid, a little tricky, a little long, because there is 3 COOH. This is citric acid. Citric acid, by the way, also is an antimicrobial. Okay, so goes here. Citric acid cycle. Next stop is isocitric acid. What means iso is this one moves. The hydroxy group is moves. Isocitric acid. Then you will say, Dr. Shi, you forgot something. Do you think I will forgot? No. This is one you learned in your chemistry class. What is that one? That's the middle one. What's it called? Cis. What is it called? Cis-asconate, is that right? We don't have that one in bacteria. Cis-asconate. We don't have that one. So we don't talk about here. In the bacteria, citric acid directly becomes isocitric acid. They jumped that intermediate chemicals. So easy, citric acid go isocitric acid. I forgot this, so we're gonna right here. Okay? This is isomerization, nothing will happen. Okay, when you see something like here, this is something, looks like something gonna happen. Okay, so what we do? Over here. CH2, CH2, COH, sorry, not this one. What's happening here? Losing CO2. NAD reduced to NADH2. This is a very important chemical. Alpha ketone, this is ketone. Glutarate. This is an intermediate metabolic precursor. It's going to go everywhere. It's going to go lipids, oxidation cycle. It's been the biosynthesis of the protein. It goes everywhere. That's called alpha ketoglutarate. Then you go here. When you see something like that, of course it's going to happen. This is a loosely attached bonds. Okay, so something's going to happen. What's going to happen? Is COOH, CH2, CH2. That's called succinyl-CoA. Again, this will happen is they lose carbon dioxide, NAD reduced to NADH2. That's not good when you connect it. So go here. That's succinate. And remember, there is GDP becomes GTP, happens right here. And I said all the time, although we talk about the ATP, but GDP becomes GTP do happens once. It's right here. That's succinate. Okay, you have a structure like this. This is very simple to understanding. That's weak double bonds. That's weak 2 there. This is easy to become this. Okay, so what we do one next one. Formaldehyde. Now, where are these goes? If you have H2 released, you're going to add something. But this time is special. FAD, add this, FADH2. That's called a formaldehyde. Okay, once your formaldehyde had a double bonds in the middle, that's not stable. Double bonds in this middle always not stable. You're going to add something there. So what will they do? They add a water in there, become malate. Do you like malate? Malate gases. That's why the beer tastes a little bit bitter, with hops, of course, malate. Okay, once you have this here, this is obviously easy to go. So later on, what will become? Oxaloacetate. Where is the H goes? Again, NAD reduced to NADH2. Then all the way back. That's what's the name we call it. Citric acid. How many ATP generate? through citric acid cycle. Then every red area we need to do. Plus, at the very beginning, that is also red. So right here, so how many we have? 3, 3, 3, 3. How many ATPs FAD become FADH2? We just say 2, okay? So this is something we want to say. NAD reduced to NADH2, it is 3 ATPs. If you talk about oxygen phosphate ratio, this is 2.5. FAD reduced to FADH2, that is 2 ATPs. If you want to talk about oxygen phosphate ratio, that's 1.5. You can either use this or use this, but not to mix, okay? So how many here? So that's 3. But don't forget, there is a substrate level phosphorylation here, so that's 1. So what's the total? Is 3 multiplied by 4, add 1, and add 1, that equals 15 ATPs. But don't forget there's 2 cycles. So 30 ATPs. So 30 here, and you get 8 here, how many you have? 38 ATPs. For a general bacterial complete aerobic respiration. Okay, now a simple question. What do you think, how many ATPs they need for a fungi going through aerobic respiration? It's going to be more than that or less than that? How many ATP going to generate if it's a fungi going through aerobic respiration? That would be 36. Now where are the 2 ATPs they go? Passing mitochondrial membrane system. Because the fungi, the citric acid is happening in the mitochondrial machinery, and you have to passing through the membrane system, which means you have to pay the ticket to go through the gate, like you pay the ticket to the parking lot. So that's 2 ATPs is energy currency. So that's why it becomes 36 ATPs. Okay, so that is the whole calculation. Okay, what is extra points for the exam? There's no space to write, I'll be right on the top, okay? If you write the whole procedure, one name is 1 point. One chemical structure is 1.5 point. You can see how many points. Okay, that's very good for the extra point. What we have so far? ATP structure is 5 points. You only write the name there is 1 point for 1. And you accompany it with structure is 1.5 points. Okay, so how many points are plenty? It's lengthy, lots of points. Okay? Okay, that's biochemistry. Now, let's say you did very good for the exam. Okay, you say, I don't need those. Because I'm not a biochemistry major. I never read the biochemistry. I don't like these lengthy chemicals. I don't like it. So, what's the requirement for this class? Then you should be at least know this. Okay, if say I don't need these, I'm doing very good. There's 2 ATPs. Here, 2 ATPs. Remember, here, 2 ATPs. Here, 3 ATPs. Here, 3 ATPs. Because NAD reduced to NAD H2. Okay, goes here. NAD reduced to NAD H2 right here. Approximately here, NAD become NAD H2. FAD reduced to FADH2. NAD reduced to NADH2. And don't forget, one GDP becomes GTP. So how many ATPs is there? 3, 3, 1, 2, 3. Can you do that? We forgot one. NAD reduced to NADH2. So it is also 3. That's good. That's for calculation, the energy. If you don't know, right there. That's very straightforward. This is old system, 2 ATPs for 3 ATPs. And this is new one. If you want to use it, it's okay. That's a new one. Okay. This 2 slides is a key for AEM 341. If you need extra points, then you just do the whole thing. And you don't need to write down the enzyme, because it's not a biochemistry class. One chemical structure is 1.0, and one chemical structure is 1.5, and one chemical name is 1.0. So that's giving you lots of the makeup. And you want to do extra points, then you need to have some effort, and this is effort. And in the exam, I'll give you 3 pages of empty paper. You can write down as many as you can. Okay, so that's what we talk about here, the key thing for this section. We can go back to review these, but something, let me finish real quick. Okay, anaerobic respiration. What we talk about is aerobic respiration on the top. The bottom of these anaerobic respiration, you can see most of these are environmental bacteria. Now, how many energies they will be generated? How many ATPs they will generate? Going through anaerobic respiration. Of course, it's less than aerobic respiration. The reason is the electron donor and electron acceptor. The distance of the voltage value is much less. So how many ATP generated? It will be somehow 36 or less. And by the way, this is a very important conclusion table. It tells you everything there. Let's look at the fermentation. What's the fermentation, the pathway to use to generate ATP? We say coupled with glycolysis. What is the final, which means terminal electron acceptor, is organic molecule. How many ATP generate? 2. How they go through? Glycolysis. So we say substrate level phosphorylation. Okay, let's look at the top. Aerobic respiration, we just talked about, spending so many times there. It's glycolysis and TCA cycle. What's the terminal electron acceptor? Is the oxygen. How many ATPs through substrate level phosphorylation? Only 4. 34 is oxidative phosphorylation. Total is 36 or 38. Anaerobic respiration, they have modified TCA cycle. Glycolysis is also modified. their terminal electron acceptor is inorganic other than oxygen. Right here, see it? 2 from glycolysis, 2 from TCA cycle, so total is 4. Substrate level phosphorylation. How many oxidative phosphorylation? 32. We skip this one because different bacteria, their cycle is different. Like I said earlier, anaerobic respiration, if we want to talk about this whole box here, this is another class, which is microbial ecology or microbial physiology. Then we go all these environmental microorganisms. Talk about the nitrogen cycle, sulfide cycle. It's all the different things. And we just skip that because we don't have much time. And that will be another 3 months we could teach, and then that's a microbiology major. So we skip that. We just tell you the total ATP is 32 through oxidative phosphorylation. Now, how many total ATP generated is 36? Okay, so that's what we have so far. What you should do when you go home, start from today. No matter how you did for exam 1 or exam 2, they are lengthy and multiple millions of extra points there, so you're going to have to study. Okay, it's very important. If you want to do makeup, that's the time to make up, and you only need to spend in time. And I said very frankly, you don't have to know everything. You know glucose. You know glucose 6-phosphate. That's already gave you 5 points there. And you know glucose 6-phosphate, you know fructose, you know that guy. This is already give you how many? It's already 10 points there. And not even goes here. Even you forget all these chunk, this you know, and this you know. And somehow, this is not difficult because it's very symmetric structure. You can remember. You remember some, you get lots of points. Okay, so that's a very nice homework. Also for you to pre-review biochemistry class. If at the same time you're taking, that's also another plus. So that's what we do. Okay, so we are going to end up right now.